Did you know your immune system might be playing a role in epilepsy? It's a fascinating and complex connection, and new research is shedding light on how immune cells and inflammation could influence your risk of developing this neurological condition. Background and objectives. Emerging evidence suggests that an imbalance in the immune system, along with inflammation in the brain, neuroinflammation, could be linked to epilepsy. However, scientists are still working to understand exactly how these factors contribute to the disease. This study aimed to investigate the potential causal relationship between specific immune cells, inflammatory proteins, and the development of epilepsy. The researchers also wanted to see if inflammatory proteins acted as intermediaries in this process. Methods. The researchers used a powerful technique called Mendelian randomization, MR. This method uses genetic data to explore potential causal relationships. They analyzed extensive genome-wide association study, GWAS, data, which includes information on immune cell characteristics and inflammatory proteins. They then used epilepsy GWAS data from the FinGen dataset. By applying 5 different MR methods, they assessed the causal effects. A 2-step MR analysis was also conducted to determine the extent to which immune cells might influence epilepsy through inflammatory proteins. Results. The study revealed some intriguing connections. The MR analysis identified 32 different immune cell phenotypes associated with epilepsy risk. Of these, 19 appeared to increase the risk, while 13 seemed to offer protection. When looking at different types of epilepsy, the researchers found 30 immune phenotypes linked to generalized epilepsy and 26 linked to focal epilepsy. Furthermore, the study found that 8 inflammatory proteins showed a possible causal effect on epilepsy. For example, some proteins like CC chemokine ligand 23, CXC motif chemokine ligand 6, CXC motif chemokine ligand 11, and vascular endothelial growth factor A were associated with an increased risk of epilepsy. On the other hand, others, such as interleukin-13, IL-13, leukemia inhibitory factor receptor, tumor necrosis factor, and osteoprotegerin, seem to offer protection. And this is the part most people miss. The researchers also found that inflammatory proteins played a mediating role, influencing between 6.3% and 13.5% of the impact of immune cells on epilepsy. For instance, CD14 plus CD16 plus monocytes were linked to an increased risk of epilepsy by elevating CC chemokine ligand 23 levels, mediating 8.5% of the effect. Conversely, effector memory double negative, CD4-CD8- T cells reduced epilepsy risk via the upregulation of IL-13, mediating 6.3% of the effect. The findings were considered robust after sensitivity analyses, P heterogeneity/pleiotropy greater than 0.05. Although none of the associations reached the level of significance after the Bonferroni correction, the results suggest that B cells, monocytes, regulatory T cells, and certain cytokines, like IL-13 and leukemia inhibitory factor receptor, are involved in the development of epilepsy, with inflammatory proteins acting as partial mediators. Conclusions. These findings provide valuable insights into the immune-inflammatory pathways involved in epilepsy and highlight potential targets for future therapies. The researchers emphasize the need for further studies to validate these findings across diverse populations and to delve deeper into the molecular mechanisms underlying these associations. Controversy and comment hooks. This research points to a complex interplay between the immune system and the development of epilepsy. Do you think this information could change how we approach epilepsy treatment in the future? What are your thoughts on the role of inflammation in neurological disorders? Share your opinions in the comments below. Source: Wu, G. et al., 2025. Immune Cell Phenotypes, Inflammatory Proteins and Epilepsy, a Mendelian Randomization Study to Investigate Causal Pathways. Neurosurgical Subspecialties. DOI.org/10.14218/NSSS.2025.00010